Okay, so A&P Lecture 18. So getting started, you've got assignment 8 due now. I assume most of you put that up on Blackboard. 9 should be there and available to you. That's due a week from Thursday. I'm doing this on Wednesday, so it's a week from Thursday on the 6th. We're also going to have an assignment. I'm going to ask you to attend the Student Success Seminar next Tuesday at 1 o'clock in Crawford, 110 Crawford. That is a test anxiety seminar. I'm under the understanding that my tests make you nervous, so this will give you some strategies to help you prepare for the next exam, which is on the 13th. Give some thought to how you want to be tested on the red blood cell path, going through the circulatory system of the bovine cardiovascular system, how you want heart events sequence tested, and blood ID, blood vessel ID, and muscle ID. Remember review sessions Monday, 5:30 to 7:30. So we've talked about sick cows. I'm going to probably move a little bit faster than I normally would. But you can pause at any time. So we've talked about sick cows. We talked about the down cows, the ones that go down and over and the problems associated with that. We've talked about acidosis. We're going to try to fit in here dehydration and DAs and a side on hypertonic saline and how that works. So we talked about acidosis, effects on cows. You have this slide. I just wanted to, and I didn't include this, and I probably should have, the acidosis, that increased VFA is going to lead to a tremendous pH drop in the rumen. The pH drop isn't the thing that really gets the cow. It's that increased osmolality that tears the rumen apart if it's severe enough. So that is there, but we got to make sure also, most people talk about low pH, but really what gets the cow is the osmolality. It shifts the direction that water wants to flow, and water can't be stopped flowing in the wrong direction, and then you get that sequence of events that eventually lead to foot problems. So want to make sure I said that. Again, water busts in, opens it up, and then all the bad things happen all the way down to the feet. So let's talk about displaced abomasums. So the thing about displaced abomasums is nobody knows how to cause them. There have been research studies where they've tried to do everything they think or know and cause a DA in a cow, and it's unsuccessful for some reason. So there, we know it's associated with low fiber diets. We know it's associated with high grain diets. We know it's usually after a cow goes off feed. We know those things, but what I'm going to propose here is a sequence of events that happens around calving that would logically explain how a DA happens. So we've got two conditions around calving that occur quite often. One is hypocalcemia. We haven't done our mineral management well enough in the dry period. The cow does not transition the way she should, and she's lacking calcium to start the lactation. So that hypocalcemia, the subclinical milk fever, leads to the smooth muscle not working. So we can think about the forestomach, the rumen, reticulum, omasum, abomasum, as not working. They have no tone. They can't contract well. So they're kind of loose and sloppy. The other thing that'll happen as we switch from a dry cow diet, which is real high in fiber, not very fermentable, to a lactating cow diet, which is very fermentable. If we haven't done our close-up transition If the cow hasn't ate well in the few days around calving, we're going to lead to an empty rumen. She's going to fill up on this rich diet, higher in grain than she's used to, lower in fiber than she's used to, and this might lead to a short-term acidosis in the rumen. So one of the things that acidosis does is we get that increase in pH. It'll also slow down the rumen a little bit. So you got two things that are stopping the rumen contractions. If we have acidosis, chances are we're not going to get the absorption of VFAs in the rumen and reticulum like we should. We're not going to get it in the abomasum like we should. One of the purposes of close-up diet is to adapt the rumen lining, the omasum lining, the reticulum lining to accelerate their ability to absorb VFA. But if the system is overwhelmed because the cow's not quite adapted yet, normally we don't get VFAs in the abomasum, the true stomach. In this case, we're going to have VFAs getting by, and when those VFAs get in the stomach, they act as a buffer. Now, normally with the abomasum, the true stomach, we want to drop the pH down to 2. But if there's a lot of VFAs hanging around, they're going to prevent that from dropping below the mid-4s. So normally you'd go down to 2. We're hanging around 4.5 or 5 because the VFAs are buffering. So one of the things that those VFAs will do, or the acid will do, is it'll drop the pH and kill all the microbes that are coming down, start their digestion. Well, if the pH isn't low enough, you're not going to kill all those bacteria. So they're going to continue to ferment. They're going to continue to do what the bacteria do. They're going to produce gas. Normally, there is no fermentation in the abomasum. There's no gas buildup there. The rumen reticulum are designed to get rid of gas, but the abomasum is not. So you've got a series of conditions here. You've got a lack of tone in the digestive tract, particularly the omasum. You got too many VFAs. Therefore, the abomasum is loose and sloppy. The bacteria aren't killed. They are going to produce gas, and that's going to lead to the stomach floating up. And as it floats up, it naturally twists. So that could be a left or right DA. Either turn there is going to lead to a twist. So to me, these are a sequence of events that could possibly happen. This is a theory on my part, but everybody I run by goes, hey, you know, that makes a lot of sense, at least for a transition cow displaced abomasum. So something to think about as we go through. Hypertonic saline. We're dealing with the body pools of the water and electrolytes in the vessels. We tend to put in that IV hypertonic saline. Which vessel are we putting in? That's one of the ones you need to know, usually. Or we may do a backup, any vein we can get it in. We put that hypertonic saline, it's got a lot more salt in the solution than we'd find in an isotonic solution. So what happens is that high dose of sodium and chloride goes into that vein, so look that one up, goes to the heart, goes to the lungs, goes back to the heart, and pumps up to the brain, where the brain can sense the salt concentration or the osmolality of the blood. So blood is tightly regulated. We now have too much sodium, too much osmolality. The brain senses that I need to drink. And because of that hypertonic solution, there's a real strong desire. As soon as that IV bottle is done, you put a 5-gallon bucket in front of a cow, it's usually gone. You usually have to go to a second bucket before she'll stop drinking. So we're talking gallons, that strong desire. And one of the benefits of that hypertonic saline is you've got a whole lot of water entering the system to try to push out that sodium and chloride that you've introduced to the system. So you're going to have that water coming in through the mouth, absorbed in the gut. Water tends to be drunk by the cow, and it tends to be diverted immediately to the abomasum. Not as cleanly as the esophageal groove does, but there seems to be a lot that goes down through the omasum and into the abomasum fairly quickly. We've got that water being absorbed. Now you've got too much water in the body. You've added 5 gallons to the blood pool. The kidneys say we've got too much blood volume. We've reduced things out. The GI tract absorption gets filtered out with the kidneys and through the bladder and then out of the body. The thing about that hypertonic saline is that acts as that big drink of water, at least 5 gallons, flushes the entire body and gets rid of some things, particularly those things associated with mastitis, leave through the kidneys. So we're flushing out the body of the toxins that are present in the animal. When they have those toxins, they tend not to want to eat, they tend not to want to drink. That hypertonic saline forces her to drink, and that allows us to flush out those toxins. We'll talk more about how that gets done in a couple of lectures. Diarrhea in calves. So there's 2 basic causes of diarrhea in our neonatal newborn calves. One is the damage of the lining of the intestines, and we lose the absorptive surface area and function. This is typically associated with a pathogen, usually viral, sometimes protozoa, sometimes bacterial, but they have it so the electrolytes and whatever can't be absorbed, so things just tend to pass through the system. Another is when we have the transport of water into the gut. So the first one is non-absorption. The second one is we have some reason why water is being pulled from the body and pushed into the gut. This is usually because of a high osmolality in the gut due to bacterial toxins or maybe poor energy nutrition. And then we have water drawn into the gut, and anytime you have a big load of water in your large intestine, you know what that leads to. We got to get it out, and it's coming out now. So we're going to lose a lot of water through that process, but we're also going to lose electrolytes, and that's the important thing. Diarrhea leads to dehydration, which is the loss of body water and the electrolytes associated with that body water. So when we're thinking replacement, we need to accelerate the water intake and make sure that we replace electrolytes as well. So you're losing a lot of water, so we need to put more water in plus the daily allotment. A cow needs a certain amount of water every day, or calf needs a certain amount of water every day. We have to put that amount of water plus some water to replace what's being lost. So it's a big consumption. So you have to think about the water in, water out, and we got to get her back into balance. So blood will be defended at the expense of all others. So we're going to lose water in the interstitial. We're going to lose water in the cells. We're going to lose water in our trans pools. We're going to lose water in the gut as it gets flushed out. They defend the blood pool at all costs because we need that transport. Without that highway working, the calf's going to die. So she's going to pull water from everywhere she can. Eventually, it will kill her if she does not have enough as she loses the water in that pool. So when we're getting dehydrated, we need water to be replaced and we need electrolytes. Sodium, potassium, chloride all need to be replaced. So we need to get those back in the body. So what do we feed our calves to replace this water and electrolytes? We usually have some sort of electrolyte solution. So this is just something we had up at the dairy. So if you look at the label here, you've got sodium and chloride, potassium and phosphate, all useful to the animal for electrolyte basis. We also have some citric acid and citrate that are going to act as an energy source to help that animal, possibly, because if she's not absorbing milk, she's effectively starving. So if we can get some energy into her as well, we can make that happen. Another thing, we can't mix these electrolytes with milk, and they should be fed separately if we can. We don't want a big slug of water and electrolytes going in with the milk because that'll mess up the milk digestion in the abomasum, where we get that clot formed, so that can be slowly digested in the small intestines. If that clot does not form, you're just going to perpetuate that diarrhea. So if we zoom in there, sodium chloride, potassium phosphate, all kind of electrolytes. We got a little bit of energy coming in from the citric acid and the citrate, but mostly electrolytes. So we want to feed those separately, and hopefully the intestines will be able to absorb that water and the electrolytes to get that calf rehydrated. So same thing again, hydro feeds, and we get in there close. They've got some lactose in there as an energy source. We've got sodium, salt, and potassium all acting as electrolytes to help get that animal back on feed. So if she can't digest milk, it's just running through her. This is a way we've got a little bit more energy in there that hopefully she can absorb the energy, the water, and the electrolytes. Again, we need to feed those separately from milk. So I would feed the electrolytes before they'll get down to the intestines, and I'd feed milk after. So maybe an hour difference there. Don't mix them together. Really. Just as an aside, what happens if we don't have a commercial product? There are other options. They are not as good. So we've got some homemade recipes that you see here up in the references up on Blackboard. Gatorade, better than nothing. It might be a little too much sugar for the animals. Powerade, any of these things here are designed to replace or work against dehydration. So if you look at these recipes, you got the homemade Gatorade here. You've got the salt, baking soda are going to be for electrolytes. The pectin is going to slow things down. The beef consomme is a little bit of nutrition as you go through. So dissolve there. This is almost pure electrolytes down here. Table salt, bicarbonate, salt substitute. You could throw those in 5 gallons of water, and you've got a mixture there that delivers electrolytes and water. You've got baking soda, light salt, or low sodium salt, that's potassium chloride. You've got baking soda is sodium bicarbonate, a little bit of pectin to slow things down, a little beef consomme to give the calf some nutrition. So different recipes there. If you don't have the commercial product or you want to try something cheaper, there, if you're willing to take the time, these are there and they are up on Blackboard as well. So we need, with our calves, to replace the water, replace the electrolytes to rehydrate the calf, And then we want to, if we can, get her a little bit of nutrition as well. So these are the references up there talking about those stowering calves and what we can do for them if you want to take the time to read them. We can also add some B complex in there. That might be some helpful things as well. Get out your letters. So what does the pulmonary artery do? Drains blood from the lungs. B, delivers blood to the lungs. C, drains blood from the rear or back leg. Delivers blood to the rear or back leg. So what does the pulmonary artery do? Pause and make your decision. What does the abdominal aorta do? A, deliver blood to the gastrointestinal tract. B, deliver blood to the lungs. C, deliver blood to the front leg. D, deliver blood to the rear or back leg. Pause and pick your answer. What does the common carotid artery do? A, deliver blood to the gastrointestinal tract. B, deliver blood to the lungs. C, deliver blood to the head. D, delivers blood to the rear or back leg. Pause and decide. What does the brachiocephalic trunk do? Delivers blood to the repro tract, delivers blood to the lungs, delivers blood to the head, delivers blood to the front leg. What are your thoughts there? External pudendal vein do? Drains blood from the gastrointestinal tract, drains blood from the mammary gland, drains blood from the head, drains blood from the front leg. Pause and give it thought. What does the portal vein do? Drains blood from the gastrointestinal tract. B, drains blood from the mammary gland. C, drains blood from the head. D, drains blood from the front leg. Give that some thought. What does it do? The cranial superficial epigastric vein do? A, drains blood from the gastrointestinal tract. B, drains blood from the mammary gland. C, drains blood from the head. D, drains blood from the front leg. Pause and give that some thought. What does the cranial vena cava do? A, drains blood from the GI. B, drains blood from the mammary gland. C, drains blood from the head. D, drains blood from the front leg. Choose your answer. Okay, we're going to talk about lungs, and we'll probably do this for about a half hour, see how far we get. And then there are some videos that complement this lecture that I've already done. Those are up on Blackboard. So we're going to start out with, here's some references to the story I'm going to tell you so you know I'm not pulling. This is Taffy. She was a cow at Michigan State when I was there. She was the world record cow, fastest cow in 2001. She was on a research trial where we studied the effect of exercise on transition cows. We found that, exercise is good for lactating or dry cows. So if you can get them up and moving, that's a good thing. But she was part of the preliminary study where we were trying to figure out whether we could train cows or not. And we got them on a hot walker and we found we can train. Taffy was a very unique cow that she just could not stop. All the other cows would give up long before she did. She had a hell of a set of lungs on her, and she just kept going and going and going and going. So when it came time, somebody took her ovaries for another experiment. So she was a dry cow. We really couldn't use her at the farm. So when it came time for auction, the equine professor at Morrisville went and bought her at auction, and then trained her at his home track. You can see that in the picture there. And then took her to the first ever fastest cow in the world contest. You can see that on the YouTube video in that link there. There was a report on it in the Farm Show magazine, if you had to get that. And she was well trained, and she was one of the few that made the whole circuit. So he got a lot of notoriety as the jockey on the world's fastest cow. But thing about Taffy is she had a hell of a set of lungs on her. So talk about lungs. We want to talk about how do cows breathe, and I'm going to ask that question over and over, and there's a number of different ways we could answer that. So the objectives, or how do cows breathe, can be answered 4 different ways. So we want you to learn the parts of the lungs and their order. We want you to learn how breathing works. What are the muscles involved? What moves? How does that get done? We want you to know the lung volumes and capacities. And then next Tuesday, we'll talk about oxygen and carbon dioxide exchanges. So all those things are, how does a cow breathe? Each of those segments is one way to answer that question. So how does a cow breathe? Let's start with the parts, are part of the lung system. So you've got an airway. It starts up in the head. You have a common entrance to the digestive tract and the airway. So we've got the nose and the mouth, the larynx and the pharynx. The trachea is number 2. And then we get down into the lungs, where we have the various parts of the lungs. So the lungs are the main organ of gas exchange, and there are a number of different muscles that are involved with that. We'll get into that in the next segment. So the airway, we have open space, very rigid, where air moves through and gets down into the lungs. We have the lungs themselves. They're right up in between the shoulder blades for our cows. And the respiration. So this is from the Atlas of Bovine Anatomy. We have the mouth and nose we can bring in there. We've got the larynx and pharynx. And then we transition to the hard trachea at the back of the head. So we have a sagittal section. We're slicing the head, putting it into left and right halves. Then we get down to the lungs. When we have the lungs, they kind of run from the first rib up front all the way over to the 13th rib. The 13th rib is where the diaphragm is attached, and it spans from one side to the other. We have different lobes of the heart. We'll have 5 different. So we have A, B, and C are the left side of the lung. We've got the diaphragm attached to the 13th rib. And then you've got E in here, that's the heart. And that's the cardiac notch. So when we're looking at our lungs, we have 5 lobes. We have 3 lobes on one side, 2 lobes on the other, because we need to provide some space here for the heart. So we've got 1, 2, 3 lobes on the left side. So we're doing a dorsal aspect. We're looking up. Nope, we're looking down on the cow. So dorsal looking down. So this is the right side of the cow, and then our left side here, the heart cheats a little bit to the left, and we got two lobes on this side. Again, the Atlas of Bovine Anatomy will give you a more detailed printout in the slide set. So the main entrance is the trachea. You have something here that's made of cartilage, fairly solid. Doesn't really change all that much. It's got to be able to withstand the pressure changes and still remain open. We branch off from there into the bronchus, individual or bronchus. The collection of them are the bronchi. So we have one here that branches into the two on this side. We have one here that supplies this lung, and then we have one here that branches into two over there. So the first part of the branch is the bronchus or bronchi, plural, and then we get down to the end, we've got the bronchioles. So all of these are more or less rigid. There's cartilage involved, connective tissue, and their job is to stay open. There's no air exchange in any of these kind of spider web things. The air exchange happens down here at the ends. So the perimeter of the cell or lungs. So we've got the branch points. Their job is to transport the air. So this is the job, looking at a human. We're looking at the ventral side or the front side of the human. We've got 1, 2, 3 on the right side, 1, 2 on the left. You got the cardiac notch over here, human trachea, bronchi, and then we get down to the bronchioles at the end. We've got the alveoli down here is where the air exchange occurs. So another human here. We're looking at the trachea. We got the bronchioles or bronchus going in either direction. We get down at the end here, bronchioles, and then we branch way down into the alveolar ducts and the alveoli here at the end. We get down to the alveoli, you have the air coming in, dividing up, and you have these grape-like structures. Each of these is an air sac. You can see the capillaries are highly interconnected. Coming in, you got the web surrounds each of the grapes. So this is what we talked about, like sheets. We're flowing over each of these alveolar sacs where the air can come in and go out. So oxygen gets absorbed, going down its concentration gradient, and carbon dioxide leaves and gets pushed back in. So another more cartoonish one of that, trachea, bronchus, and then the bronchioles down to the duct system. Again, we have the alveoli here. Another diagram here. The red lines are representative of the capillaries. We have an artery feeding those capillary beds, and then the vein taking it back. The colors are wrong in this case. The artery should be blue. The vein should be red. So we have all these hard bronchioles, and then they go to these collections of sacs down here that sort of look like grapes. These are the things that will expand and contract. The bronchi here, the bronchiole, will not. They also have smooth muscle fibers surrounding them so they can open and close. Those muscles contract, they'll collapse the bronchioles. That's where we get into asthma, when those things get overexcited and close things down, or anaphylaxis. But they're mostly rigid. They don't close all the way. The big thing here is these alveolar sacs at the end can expand and contract greatly. Another view of that, we've got the bronchiole coming down, pulmonary artery, so this is the right color scheme, and then the veniole going back. We have those sheets of interconnected capillaries bathing these alveolar sacs with all their little bubbles in them. The air moves in as they expand, and then they, as they contract, they move the air back out. So if we're looking at the order here, we start with the mouth or nose, the pharynx and larynx next, the common trachea in the neck. Once we get into the lung tissue, we have the bronchi, we have the main, the lobular, and segmental, getting smaller and smaller. Then we transition to the bronchioles. All of these in black are involved in just moving air. They are not involved in the gas exchange. The gas exchange occurs down here in the alveolar ducts and the alveolar sacs. So this is where oxygen is absorbed into the body, and then carbon dioxide comes out, and then as the alveolar ducts and sacs contract, that carbon dioxide is pushed back through the bronchioles, bronchi, trachea, larynx, out the nose and mouth. So those are all the parts. We can ask the question again, how do cows breathe? Let's talk about the mechanics of that. So breathing muscles, we're going to talk about the diaphragm. That is our major inspiratory muscle. The diaphragm contracts, it gets shorter, and pulls the lungs open. So we'll show you a picture of that in a minute. You have the external intercostals. They assist in moving the ribs and bringing air into the body. So trying to open up the ribs, increase the space, and bring air in. The internal intercostals are used for forced expiration. So expiration in this case means breathing out. So they're going to try to close down the rib cage and move air out, squeeze air out. The abdominal muscles can help with forced expiration. So if those abdominal muscles contract, they are going to push on the guts, which is going to push up in the diaphragm, push the diaphragm up into the rib cage, and squeeze those lungs down. So we have the diaphragm sits down here at the bottom. You got the muscles, the intercostals are going to line the inside and outside of the ribs. You're going to have the lungs are going to be stuck to the very smooth inside of the thoracic cavity. They're going to be stuck there by basically water. So there's a surface tension that holds the lungs up against the ribs. So as the ribs move, the lungs move. Is when we get space introduced in there that our ability to breathe is compromised and we get a collapsed lung. We can think about the lungs stuck to the ribs as kind of two plates of glass with water in between them. You can slide the plates of glass around, but you can't pull them apart. And that's how, if you like lift up one piece of glass, the other one will remain stuck to it as long as there's water there. And that's how the ribs are connected very loosely to the lungs. We need the ability to slide up and down. We need the ability to move. Tacking the lungs to the ribs will limit our ability to breathe. So as the diaphragm tightens up, it's going to flatten out, it's going to move down, and it's going to pull the lungs down. It's going to expand these alveoli and draw air into the system. So you've got that space there, the pleural cavity filled with a liquid that's going to hold the lungs to the smooth wall that's inside of the ribs. Another view of this, you've got the ribs, you're showing the external muscles, external on the outside, internals deeper in. The externals are going to basically work the spinal cord and try to open up the ribs by pulling either side out, and we'll get some stretch in the connective tissue that connects the ribs to the sternum. So it's going to open it up like a clam. And then the intercostals, internal intercostals, are going to pull things back. So those are in opposition. So internal intercostals right there, external intercostals out there. They're going to work. So we got to remember that everything moves from a high to a low. So we have a resting stage here. We go from resting to inhale to exhale to rest, inhale, exhale. The resting might be a little short sometimes if we're really working out, but you have that sequence. So rest, inhale. So the diaphragm is relaxed. It's attached to each side of the ribs. We have the lungs attached to the wall, and we're kind of elastic snap back. Okay, in the second stage, we have the B, inspiration. So rest, inspiration. So the diaphragm's a muscle. It contracts and flattens out. While it does that, it pulls the lungs down. We also have the external intercostals pulling the chest wall out. So you're expanding outward with the ribs, and the major inspiration comes from the diaphragm moving down. We're expanding those lungs. With this expansion, you're going to have a slightly negative pressure inside the lungs. So air is going to move from a high to a low. The air outside your mouth is higher pressure than inside your mouth. Not a big difference, but enough to move air in. So the air is moving from a high pressure to a low pressure. Then we have the expiration, the exhale. The diaphragm relaxes, the lungs snap back, so the diaphragm moves up. The internal intercostals will collapse the rib cage, so we're starting to squeeze on these lungs. We can also contract our abdominals and help do that. Now, the air in the lungs is higher pressure than the air outside. Everything moves from a high to a low, so air is going to leave the lungs being pushed out, high pressure to a low pressure. So you can kind of see this side by side where everything is. So inhalation, diaphragm comes down, the rib cage, the muscles move things, stretch the cartilage here, move stuff up, lower pressure in the lungs, air moves in. Diaphragm moves up, relaxes, the intercostals close down the rib, higher pressure in the lungs, moves the air back out. The major movement is by the diaphragm. They're going to pull by that surface tension. They're going to pull the lungs down and expand their volume. And then when it relaxes, everything snaps back and contracts and pushes the air out. So breathing muscles, you should know what all of them do by now. So diaphragm helps us breathe in, external intercostals help us breathe in, internal intercostals and the abdominal muscles help us breathe out. So thinking about snorkeling, If we look at snorkeling, when you're out there swimming around and snorkeling, it's very hard to breathe. And there's two reasons for that. One is now you've increased the length basically of your trachea. So that means you've got that much more dead air that you have to move in and out. You have a longer pathway to move air before it can get to the alveoli where gas exchange occurs. So extending that length means you're going to have to work harder to move that air in and out. So if you just start breathing through that pipe, you'll notice that you're working harder to move air in and out to get the gas exchange that you need to. It's also going to require more effort to move that air because you've got more resistance, more distance to move everything. So your straw is much bigger. So not only is the exertion of swimming, but even if you're just laying there on the surface of the water, you're going to find that you're going to be breathing a whole lot harder because that snorkel is extending the dead space and making it harder for us to get air in and out. So something to think about. We also, there was a woman, she was one of the first to swim across the English Channel back in the days of yore. If we had this up on the projector, you could read it, but it talks about as a swimmer, she had a remarkable ability to expand her lungs. So they talked about some of the great athletes that were her peers at the time. Boxer, I think, was one of them. Their ability, they could expand the circumference of their lungs only about half of what she could. I can't read that right there, but if you zoom in on it, she had the tremendous ability to expand her lungs, and that's what made her such a great swimmer and gave her the ability to get across the English Channel pretty much before anybody else. So next thing we're going to talk about is lung capacities and lung volumes. What I want you to do is go up and look at the two YouTube videos up on the Blackboard. You go to the content folder and then the lungs folder. That'll give you an animation of what I'm talking about in the next few slides. We'll also give you that syringe next Tuesday. I probably should have given it to you last Tuesday, but I forgot. So we'll give it to you next Tuesday to learn how that all gets done. So it's a hands-on visual representation. I filmed it, it showed it to you. We'll discuss it here. So how do cows breathe? Here's the third way we're thinking about it. There's 4 lung volumes that we need to think about, and there's 4 lung capacities that we need to think about. So the 4 lung volumes, these are mutually exclusive. They don't overlap. The 4 here will add up to our total lung volume. So the one we usually think about is our V subscript T, or tidal volume, that changes in volume of the lungs associated with normal breathing. If we need more air in, we're going to use up our inspiratory reserve volume. So IRV. Our ERV, if we need to blow out some candles, especially at my age, there's lots on the candle or birthday cake. We're going to use our expiratory reserve volume, the additional we could breathe out if we needed to. And then there's the residual volume. That's as small as we can make our lungs from blowing out, collapsing our rib cage, letting the diaphragm and the abdominals push up. That's as small as we can make our lungs while we're living. So that's the residual volume. If we look at it, an adult cow, we can stack those volumes up. We have the residual volume, RV, at the top. That's about 6 liters. We've got the expiratory reserve volume next, about 6 liters. We got the tidal volume in our cows, about 4 liters, and the inspiratory reserve volume, about 6 liters. So a gallon and a half, 1 gallon, gallon and a half. So we're looking at about 6 gallons, 5-6 gallons of capacity for the lungs of our dairy cows. We can think about the capacities as a combination of the various lung volumes. The first capacity we'll talk about is the vital capacity. That's VC. IC is the inspiratory capacity. The FRC, functional reserve capacity. And then TLC, in this case, will mean total lung capacity. How do they work together? So here's our volumes stacked. We told you the volumes never overlap. With our capacities, we have some overlap. So vital capacity is the air we can move in and out of the lungs. It's our expiratory reserve volume, tidal volume, and inspiratory reserve volume. This is the amount of air we can move in and out. That's about 4.5 gallons. Well, it adds up there, I think, to 4 gallons total right there. Over here, TLC, we have the total lung capacity. That is the sum of all 4 volumes. And then we can add up total lung volume or lung capacity, functional reserve plus inspiratory capacity. How much can we breathe in? After normal here, those two are going to add up to all 4 volumes. Functional reserve, residual volume, expiratory reserve, inspiratory capacity, tidal volume, and inspiratory reserve volume together. So writing that out, vital capacity is the air we can move in and out, tidal volume plus inspiratory and expiratory reserve volumes. Inspiratory capacity, tidal volume, and then whatever we can breathe in above that normal breath. Functional reserve is our other two, residual volume, expiratory reserve volume. And then total lung capacity is all 4 volumes added together. So here's the definitions in case you need them. So explaining what each one is. So you can pause that and go through. It should be in your handout. So you can also have a video here if you want to look at a YouTube video that talks very well about that. So that's an additional reference that you have. So I have the volume stacked, but this is probably a better way of looking at it from Duke's Physiology, the 12th edition. You have a little circle here, which is the residual volume. Over top of that, we can generate the expiratory reserve. We have the tidal volume in a circle there, and then we have the full lung capacity is kind of the volume of air that wraps around that other. We have some here, some tracings. We'll look at some other ones that are a little bit better, but this shows you how the apparatus was put together. We have a little dog here with a gas mask on. It's going to breathe in and out of this cylinder. As it exhales, it's going to breathe in and out. It's going to move this cylinder up and down. So as it breathes in, the wheel here is going to drop the pen that's tracing. So we'll get down to maybe a reserve volume. As it breathes in, pulls the string down, the pen goes up all the way up to the possible potential maximum volume that we can have. So that's how the system works. Breathing in, breathing out, and then tracing. Here are some better diagrams illustrating that. So what's the diagram? So we're breathing normally here, tidal volume. So breathe in, breathe out, breathe in, breathe out, breathe in, and then we breathe in as much as we possibly can. And then we let our lungs relax and we go back down to the demarcation of the tidal volume. Breathe in and then blow out as much as we can. Breathe in normally, breathe out, breathe in, and then try to blow everything out as much as we can. And then back to tidal volume over here. So as we make our changes, we go from normal tidal volume, we add our inspiratory reserve volume there and there. That becomes our inspiratory capacity. As we breathe out, we've got our tidal volume, we're pushing that out, as well as our expiratory reserve. We can only go down to a minimum lung volume of our residual volume. We go from the bottom of our tidal volume, breathed out. What's left in the lungs is our functional reserve capacity. If we breathe in as much as we can and then blow out as much as we can, like we do when we're blowing out candles on a birthday cake, that's going to be our vital capacity. And our total lung volume, again, is all four volumes put together. So we got arbitrary time passing while a patient is breathing in and out of a tube, and we're measuring the airflow in and out of that tube. So volume of the lungs in here, this is the maximum volume we can have in the lungs up top here. Down here is the minimum volume we can have at residual volume. Another diagram showing the same thing. The big difference here is we're starting to exert and we have the air moving in and out, expanding, and it's stealing from our inspiratory reserve. And this is the air moving in and out, stealing from our expiratory reserve. So we start to exercise, we're going to expand and contract our lungs much more. I think the next diagram does that as well. So same thing, we've got the inspiratory reserve up here, we've got the tidal volume, we've got the expiratory reserve, the residual volume down here. As we start to exert ourselves, we start breathing faster and faster, moving much more air until we've reached our maximum capacity. As much air as we can get in, as much air as we can get out, that total range. We'll see that with our showmanship heifers as they get loose. They'll be running around, and at some point they can't run anymore because they can't get any more air in. They've used up their total or their vital capacity, using all they can breathe in, all they can breathe out, trying to get more air in, and they'll stop on the hillside and look at you and be breathing heavy. So again, those syringes, we'll give those out next Tuesday. You can go up on Blackboard in the course content folder, the lungs folder, and see some videos of me using them. What I'm doing is using a syringe to simulate a lung. It's a hands-on approach to what we're doing. So we got about an hour here of content. There's probably 10 minutes in those other videos, and I probably talked a little bit faster than I expected to. So if there's anything that's unclear, bring them to Tuesday's lecture, and we can go over those 3 different ways to answer the question of how does a cow breathe? What are the parts that she breathes with? How does the mechanics of breathing, and then the capacities and volumes that we deal with. So we'll end there and get this posted.